नहीं थोड़ा ऑनलाइन में 2-3 साल से कर रही हूँ घर में, बहुत आदत हो गई। Yeah. Hello students, welcome back to Botany first year's class. So we have already done with almost all FAQs from all chapters except the 9th and the 10th chapter. So we will see a quick glance over there so that we can finish it off the SAQ part. Then we can start the most important VSAQs where you have to get 20 marks. So we will see there also. Okay. Now mitochondria structure was over, functions were over, nucleosome, cartwheel structure of nucleosome was over, centriole was over, cartwheel structure of centriole was over, and the chloroplast, mitochondria was over, cell theory also over. So what else? SCR, rough endoplasmic reticulum, smooth endoplasmic reticulum was also over. Now only 2 questions left out in this chapter. What we have to go, that are the give a brief account on the chromosomes of centromere. This is the one of the questions. Yeah. When we are saying the what is the based on the position of the centromere, different types of chromosomes will be there. So what is meant by centromere? Centromere is the part that is present at dividing the chromosome into arms. This part is called as the centromere. So this centromere is the non-stainable part. Centromere. Centromere is the non-stainable part of the chromosome which connects the 2 chromatids or which divides the chromosome into chromatids. Okay, that is called as the centromere. And this centromere will be present at different positions on the chromosome. Based on that, we can say 4 different types of chromosomes are there. The first one is the metacentric chromosome. Metacentric chromosome. Metacentric chromosome. So in this, where the centromere is present, centromere is going to be present. Wait, I draw here only, metacentric. Exactly at the center of the chromosome, so that it can divide the chromosome into 2 equal parts, 2 equal chromatids. Okay, 2 equal arms. These are the arms of the chromosomes, and this is the centromere. When centromere is present at the center of the chromosome, dividing the chromosomal arms into 2 equal sides, that is called as the metacentric chromosome. During the cell division, that is at the metaphase and at the mitosis and the meiosis stages, where this is metacentric chromosome gives the shape of the V, capital V shape. Okay, and now comes the submetacentric chromosome. In the submetacentric chromosome, in this what happens? The chromosome is going to be divided into 2 unequal sized parts, 2 unequal sized arms. This is the centromere. And this is the arm. And this is the another arm. 2 unequal sized arms are going to be present for the chromosome by the presence of the centromere at one pole of the moving to one pole of the chromosome. That is the submetacentric. During the cell division, it gives the J shape. It gives the J shape during the cell division. Okay. Next comes the acrocentric chromosome. The acrocentric chromosome means when the centromere is placed acrocentric chromosome. When the centromere is placed at one pole of the chromosome, making the dividing the chromosome into two unequal sized arms. One is very long arm and another one is the very short arm. Like this, whenever the chromosome was divided, that is called as the acrocentric chromosome. Okay, arms. And this is the long arm and this is the short arm. Okay, that is the acrocentric chromosome. Now, and during the cell division, it makes the, during the cell division, submetacentric chromosome gets the L shape and the acrocentric chromosome makes the J shape. Okay, and the telocentric chromosome. Now comes the last one is the telocentric chromosome. Telocentric chromosome is the one which makes the where the centromere moves to exactly to one pole of the chromosome, which is dividing the chromosome into like this in such a way that the here one small dot will be left out. Otherwise, only the chromocentromere is completely moving to one pole. That is the telocentric chromosome. And during the cell division, it gives the small I shape. Okay, like this, we have to say about the position of the centromere and the chromosome. This is a very simple one, and we can get easily 4 marks for this. Like this questions we can expect for the supply paper. Okay, now comes the last question from this is the nucleus. Describe the structure of the nucleus. Okay, in this one more thing. Now only just I remembered to say you. Whenever in botany any question comes, then you have to, if the diagram is there, you need to draw the diagram. Here, we won't suggest that you have to draw the diagram. We won't say anywhere, we won't ask in the question, but you need to draw the diagram for in this subject itself. Now comes with the next part, next question, that is the structure of nucleus. Describe the structure of nucleus. Okay, so whenever we are saying the nucleus, where it is present and how it looks. Okay, nucleus is a double membranous cell organelle Nucleus is a double membranous cell organelle, and the two membranes are not present in a continuous manner. That is, there are some interruptions in between the two membranes. That is the nucleus. Okay, and inside this, it is having the genetic material that is present as a chromatin in it. In the nucleolus, and apart from that, it is having the Okay. And now what we have to say, nucleus is also, it is present in the cytoplasm, which is a constant, which is a circular shaped cell organelle, which is having the two membranes. The cell organelle is having the two membranes, and complete nucleus, we can say nucleus is controlling the complete behavior of the cell. We can call the nucleus as a cell brain. Nucleus as a cell brain. Okay, and this nucleus was first identified by Robert Brown, and this nucleus is having the totally 4 parts in it. That is the nuclear membrane, nucleoplasm, chromatin, and the nucleolus. Okay, so first one is the nuclear membrane. Nuclear membrane is a double membraned one, which is present, which are smooth ones. Two membranes are the smooth, but those are the discontinuous. That means they are leaving a small gap in between the two membranes. So this is the outer membrane. And this is inner membrane. Nuclear membrane is made up of 2 layers, outer and the inner smooth membranes, which are not continuous, and from the outer membrane, which is having the ribosomes on its surface. So this is called as the rough endoplasmic reticulum. Yesterday we discussed about the rough endoplasmic reticulum. Now, this is the one which is originated from the outer membrane of the nucleus. Okay, these are the yellow ones, are nothing but the ribosomes on the surface of the endoplasmic reticulum. Okay, and wherever the gaps that are present in between the nuclear membrane, those are called as the nuclear pores. And these nuclear pores are meant to make the easy passage of the proteins. Those are prepared by the genetic material inside the nucleus, as well as transfer of the genetic material at the time of the need. When the need is there, at that time it can transfer easily the genetic material through these nuclear pores into the cytoplasm. That is the main purpose of the nuclear pores. Okay, those are present in between in the nuclear membrane. And now comes this is the rough endoplasmic reticulum. Rough endoplasmic reticulum. Now comes inside the nuclear membrane, a dense semi-solid gelatinous material will be present, that is called as the nucleoplasm. This is called as nucleoplasm. And this nucleoplasm, now comes the second point, nucleoplasm. Nucleoplasm is a substance that is present below the nuclear membrane, which is very rich in the nuclear enzymes. Very rich in the nuclear enzymes. Very rich in the nuclear enzymes, which are meant for the preparation of the genetic material and for the further doing the RNA technology or for anything. The genetic material related nuclear enzymes are present in the nucleoplasm, And apart from that, in the nucleoplasm, RNA molecules are present. These are the RNA molecules. Okay, RNA molecules are present and some proteins are also present in the these are the proteins that are present in the nucleoplasm. Okay, then we have to go with the, and these are very rich in the enzymes. Which type of enzymes? The RNA polymerase, DNA polymerase enzyme, and the ligase enzyme, like that. And the next one is the chromatin. Inside the nucleoplasm, a darkly stained structure is present called as the nucleolus. This inside the nucleolus, a thread-like structure will be present. That is nothing but the chromatin net. That is nothing but the chromatin net. This one. This is the chromatin net. And this chromatin net during the cell division, which condenses to form the specific number of chromosomes according to the organism. Because the chromosomal number will vary from organism to organism. According to that only, chromatin net condenses itself to make the chromosomes in the organism. Okay. And the last one is the nucleolus. And this nucleolus is the one which is present in the nucleoplasm of the nucleus, which makes the chromatin net and the other enzymes and the proteins which are necessary for the organism to have during their cell division, as well as for their organelles formation and etc. And the next one in this is the everything was over from this chapter. Now we will go with the 10th chapter, that is the biomolecules. So in this biomolecules, what are the questions what we have to learn? So in this, they may ask that the schematically represents the primary, secondary, and tertiary structures of the hypothetical polymer using a protein. This is one of the question what we can expect. And the another question will be the nucleic acid exhibits a secondary structure. Justify. Nucleic acids exhibit secondary structure. Justify. Okay, so only these two questions we need to learn over here, and the VSQs are mandatory because one chapter, one VSQ. Okay, so the first one we'll see this most important one. Nucleic acids exhibit the secondary structure. So nucleic acids are the ones which are having the secondary structures mostly in the organisms, in any organism. Because secondary structure means the primary structure which is present, which will make the another bond with another nucleic acid, that is called as the secondary structure. Okay, so the secondary structures of the nucleic acids can be exhibited by the DNA molecules. These are going to be exhibited by the DNA molecules. Okay. This was given by Watson and Crick. Given by Watson and Crick. The secondary structure of DNA is the nothing but the secondary structure of the nucleic acid. Okay, and this will be explained with the double helical model of the DNA, which can be explained with the double helical model of the DNA. Like this, the double helical model will be present. We'll take another strand with another color. Okay, this is the 5' to 3'. Here it is a 3' to 5'. So this is the double helical model of the DNA strand. And the two strands of this are anti-parallel to one another. Like this, the two strands of the DNA will be present anti-parallel to one another. That is, if one strand is moving from 5' to 3' direction, and then the second strand will move from this side. This is the 5' to 3'. Okay, that is the anti-parallel to one another. And these are present in the opposite direction, that also in the opposite direction. And the backbone of these two is the phosphate group. Okay, just like the, see here, it is having the linkage at the back, that is the phosphate is the making the backbone of this genetic material. Okay, phosphate makes the backbone for the genetic material, and this backbone is having the link between the sugars, sugar, phosphate, and sugar. Okay, so in between the two pentose sugars, one phosphate group is going to be attached like this. This is a pentose. Pentose sugar means which is having the 5 carbons in it. That is the pentose sugar. When this pentose sugar is having a phosphate bond, that is at its back, that is nothing but the sugar, phosphate, sugar. Okay, that is the one what we can say as the backbone of the double helical model. And in this, the nitrogen bases will be projected outside. And these are perpendicular to the phosphate group. Those are present. Okay, so that is that are facing inside. Okay, so the phosphate groups are facing inside, and these nitrogen bases are facing inside, and the phosphate group is outside. So that is present as the inside for the phosphate group. And in this nitrogen bases, adenine, A represents the adenine, and G represents the guanine, and T represents the thiamine, and C represents the cytosine. Okay, like this, the 4 nitrogen bases are present, which are represented with AGTC, adenine, guanine, thiamine, and cytosine. And in this, adenine always makes a bond with the thiamine with a double bond. Adenine always makes a double bond to the thiamine, and guanine always makes a triple bond to the cytosine. So that these bondings are completely made up of the hydrogen bonds. These are hydrogen bonds. Okay, like this, the nitrogen bases are present, and all these are present in the double helical model of the DNA in a stepwise manner. That means in one helix, nearly otherwise approximately 10 nitrogen bases are going to be present. Okay. Like this, in one helix, nearly 10 nitrogen bases are going to be present, just like a staircase. Okay. And each step of ascent is represented by a pair of nitrogen bases. Whenever the two strands are there, then the nitrogen bases are present, means that is in a pair. In between the two strands, when the nitrogen bases are present, those are present in a pair. Okay, and in each step, and the one helix is having the 360 degrees. One helix is having the 360 degrees. In each and every step of the ascent is having the 36 degrees. And the pitch width will be 34 angstroms. 34 angstroms. And the distance between the two base pairs is the 3.4 angstroms. Because in each and one helix, 10 base pairs are present. What we said, in one helix, ten base pairs. So one helix is of 34 angstroms, and the distance between the two new nitrogen bases will be 34 by 10. That is 3.4 angstroms. In each and every nitrogen base, it is having the 3.4 angstroms is the distance between the each and every nitrogen base. And with this, it can make the double helical model very tightly, and which is undisturbed. The double helical model is the very strongest double helical model of the DNA. And this is the, and in the DNA, we will be having. So this is the right helical model of the bDNA. Yes, that is a very rarely they will ask primary, secondary, and tertiary structure of the protein. Okay, of a polymer using a protein. Give an example. So the protein is having, the proteins are completely made up of the phosphate groups, which are having the 4 levels. That is the primary, secondary, tertiary, and the quaternary structures. Okay, these amino acids are present in the all the amino acids that are present in the organism are making a chain-like structure to make the specific proteins in the organism body. Specific proteins in the organism body. So in that, the first amino acid, and in a chain-like structure, whenever the amino acids are present, we can call them as the first amino acid and second amino acid. Like that, we can give the number for the amino acids. And here, like this, how many amino acids can make a chain-like structure? Total 20 different amino acids can make a chain-like structure to form a protein in any organism. So that is called as the primary structure, which does not have any branches in it, and only the amino acids are arranged in a chain-like manner, simple chain-like manner. That is the primary structure. If the this protein structure is having any branch, that is the ended up, and the first amino acid is, and in this, the amino acids are having the N terminal and the C terminal. Okay, the first amino acid is having the N terminal, that is the present at the terminal, which is having the N. Okay, that is the first amino acid. And the C terminal is the end of the protein chain. Okay, primary structure, where the amino acid that is present is called as the C terminal amino acid. And always these proteins are having the right-handed helical structures. Right-handed helical structures, just like the DNA. Okay, just like the DNA, even they are having the right-handed helical structures. And these are present as a proteins present as a thread-like structure, which is folded. That is called as the secondary structure. Okay, and the next one will be the tertiary protein structure, which is having a chain-like manner, which is having the many folds in it. That is called as the tertiary structure of the protein. This much we have to write, and we can end up this question. Okay, so this is a very rarely they will ask, but we cannot take a chance right now. So we have gone through this. Now we will go with the VSQs from all the chapters. So shall we start with the last chapter VSQs? Yeah, last chapter, that is the ecology. Because from this chapter, one VSQ is mandatory. Out of 8, one VSQ we will get damn sure from the ecology chapter. So what is the first one? The climax stage is achieved quickly in secondary succession as compared to primary succession. Why? Because the primary succession will be done on the first time on the soil, that is the plant growth. Secondary succession will be done from that. So soil is already there when the secondary succession is going to be starting. So secondary succession is faster than the primary succession. Okay, primary succession means where the plant life need to start. It means they need to struggle hard to make their population, their community more over there. So for that, the primary succession will be late enough than the secondary. So why the secondary is faster? Because soil molecules with the nutrients is available for the further growth of the otherwise increase of their number. So the secondary succession is very fast to reach the climax. Okay, next one. Among the bryophytes, lichens, ferns, which one is a pioneer species in their succession? In their succession, lichens are the pioneers in the xeric succession. Okay, in the xeric succession. And the next one, the most important one, from here we will give any 2 examples of the xeric succession. They can wind up like this. Lichens and the bryophytes are the best 2 examples for the xeric succession. And the next one, name any type of land plants that can tolerate the salinities of the sea. Those are the halophytes and the rhizophora. Halophytes, and the example is rhizophora. Okay, these plants which can grow in the saline water, otherwise they can tolerate the salinities in the water. Those are the halophytes. Example, rhizophora. Next one, define hydrophytes. Define heliophytes, sorry. Define heliophytes and the steophytes. Name a plant from your locality that is either heliophytes or sciophytes. Okay, so now in this, what we have to say, what is meant by heliophytes, what is meant by sciophytes, we have to say. The plants which grow in the direct sunlight. The plants which grow in the direct sunlight are called as the heliophytes. Okay, so those are the example, sunflower ficus, which we can say very easily now. Ficus means fig. Okay, sunflower also we can say. Easy to remember. One example is mandatory. And the next one, sciophytes. Sciophytes means the plants which are growing in the shady areas. The plants which are growing in the shady areas are called as the sciophytes. Example for this, funaria, mosses, club moss. That is the funaria. And the next example, next question. Define communities and the population. So in this, we have to give the definition for the population as well as the community. What is meant by population? A group of similar individuals. A group of similar individuals belong to the same species. Belong to the same species and found in growing in an area specific area are called as the population. Okay, population means a group of similar individuals belonging to the same species are growing in a specific area are called as the population. Next one is the community. What is meant by community? Assemblage of the populations of different individuals is called as the community. Okay, and the same thing we'll be having in the next question. Define communities. Who classified the plant communities into hydrophytes, xerophytes, and the mesophytes? Define communities. Communities means the assemblage of different populations of the different organisms growing in a specific area are called as the communities. Who has classified the plant communities? Eugene Warming. Eugene Warming. Classified the plant communities into hydrophytes, mesophytes, and the xerophytes. Okay, next question. Hydrophytes show reduced xylem. Why? Because they are present in the abundant water medium where the plant body does not need to absorb and pump the water. So xylem shows reduced nature. This much you have to write. So in this, what are the important questions what we can expect for the exam? Hydrophytes show reduced xylem. Why? This question. And define communities and who classified the plant communities into the hydrophytes, mesophytes, and xerophytes. Define population and community and define heliophytes and sciophytes. Name a plant from your locality that is either sciophyte, otherwise a heliophyte. Okay, so like that, 4 questions out of 8. If you concentrate on the 4, the other 4 also just read, but concentrate on this, 1 will come. That much guarantee. That means 2 marks in this, and the 1 question is the SAQs, 6 marks total from this chapter. And the next comes the cell division. In the cell division, because the 12th chapter anatomy, from that chapter we won't get any VSQ. No need to learn that chapter VSQs only. Only SAQs, LAQs are the important ones from that chapter. Prepare with the SAQs. Okay, now go with the 9, 10, 11. That means 3 chapters, 3 VSQs is mandatory. Okay, so the first one in the cell division. In this also, what is the most important one? So the give the average duplication time of a E. coli in 20 minutes. How much time it will take the cells to divide? Okay, So here it is taking completely 80 minutes because give an average duplication of a cell in E. coli in 20 minutes. One E. coli will go with the division in 20 minutes. That's the one what he has given. And how much time it will take to go with the here two cells. He has asked with the how much time will two E. coli cells will become 32 cells. Okay. So for making the 32 cells, it takes 80 minutes. How? Because two cells will undergo mitotic division to form 4 cells. That is in the first 20 minutes. Next 20 minutes, 4 will become 8. And in the next 20 minutes, 8 will become 16. And in the next 20 minutes, 16 will become 32. That means how many 20 minutes are there over here? 1, 2, 3, 4. That means 4 2s are 8. 80 minutes is the answer. If you write 80 minutes, done. 2 marks you will get. Okay. But here the question may change. That means they are asking with the 2 E. coli cells which starts the division over here. Sometimes they may ask with only 1 E. coli cell. Read the question and give the answer. Don't mug up the answer. In this, twist will be there. Okay. What is the one? They are saying that E. coli cells will undergo division in 20 minutes. And the division starts with the 2 E. coli cells to become 32. Then 80 minutes is the time limit will be consumed. If one cell starts to become 32, then that is the 100 minutes. Increase one more 20 minutes. 1 to 2. 1 becomes 2. So like that, we have to use the brain over here. Okay. They may change the numericals. Otherwise, they may change this number 32 cells instead of, they may say 64. Then increase the number. Okay. 20 minutes. Like that. Next comes the, like that one more. In a tissue, if a tissue has a given time 1024 cells. Read or see in your materials. If a tissue has the original parental single cell undergone. One single cell they ask. So how to do this? One cell has to become 1024 cells. That means how many times it will undergo the mitotic division? That means one cell will become 2 cells in one mitotic division. 2 to 4, 4 to 8, 8 to 16, 16 to 32, 32 to 64, 64 to 128, 128 to 256, 256 to 512, 512 to 1024. So how many times the division occurs? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 times the mitotic division occurs to become one cell to 1024 cells. 10 times. And same thing, they can change the number. They can ask that one cell will become 256 cells. How many mitotic divisions will occur over there? Less than 2 divisions. That is 8 divisions. If you remember this, they may ask between any number. Starts with only one cell over here, but they won't start with the 2 cells. If they are starting with one cell, 8 divisions to make the 256 cells and 9 divisions for the 512 cells. Like that, we have to say this answer. Only the number, if you write, that's sufficient to give the 2 marks. Okay. And only 2 minutes. Left out one question, I will finish. The answer is having 1200 pollen grains. How many pollen mother cells must have been there to produce them? So when this type of question is there, then what to do and how to do? Very simple question. Even we can do the calculation without the paper and pen. But here I need to show. So here what they are saying, 1200 pollen grains are there. And they are asking how many mother cells. One mother cell, one microspore mother cell by the meiotic division produces 4 haploid cells. So here what they ask, 1200 pollen grains, haploid cells means pollen grains. Then how many mother cells? That means 1200 into cross multiplication beta. 1200 into 1 by 4. Otherwise simply 1200 by 4 also you can do. 4 3s are 12. 300 microspore mother cells will be present over there to make 1200 haploid pollen grains. Okay. So like this, they can change the number and they can ask you. So be prepared with these questions, and we will time was up so that I cannot make the many more questions over here. So tomorrow we will continue with these important chapters where the numericals are there, where you can get 2 marks as it is. So stay tuned to the same channel, same